When talking about low-grade glioma biology, it immediately comes to mind that it will be much easier to focus on one of the many diseases that are contained in low-grade glioma. So I will start talking about kind of a more homogeneous and well-defined group, so-called polycytic astrocytomas. And then also try to cover all of the remaining things that are sub-summarized under low-grade glioma at this stage. So polycytic astrocytoma, until about 5 or 6 years ago, the only thing we knew was that it was frequently or sometimes associated with NF1 disease. And that we definitely needed the surgeons to cut it out, because then we have a very good progression-free and of course overall survival. We also knew that there were some age-specific differences in survival, which we now think are not only related to different histopathology, but also mainly to tumor localization, and of course also to biology. And then, as I said, about 5 or 6 years ago, it started to really become more exciting, because then it started to become more biological. And the first thing that several groups in the world noticed was that when looking at copy number aberrations in polycytic astrocytoma, there was only one really interesting thing, which was a tandem duplication occurring at chromosome 7q34. And quite a smart guy back then, working in Cambridge with Peter Collins, called David Jones, whom you will probably hear several times at this meeting, basically figured out that this tandem duplication on chromosome 7q34 leads to a fusion of the kinase part of BRAF with previously unknown or uncharacterized other genes. And we now know that this is really the hallmark genetic aberration in polycytic astrocytoma. We know that this can be caused primarily by this duplication, where we then get these two bits duplicated and then put right next to each other, and then get this fusion gene, which basically, and this is common to all these fusion genes, is that it retains the kinase domain, which is basically the part of the BRAF gene that is driving oncogenesis and cell proliferation, and it loses its autoinhibitory domain, which usually is responsible to prevent activation of the gene. We also know now that alternative fusion genes can be caused by deletions at that same locus. So we have already known before we even started to do whole genome sequencing on these tumors that quite a substantial fraction of them, and I think it's probably around 85%, have hits in one single pathway, which is the MAP kinase pathway. And you can just see depicted here that these hits are mainly in the BRAF gene, but of course we have these NF1 patients, which is also a gene that is feeding into the MAP kinase pathway, and we have rarer mutations in KRAS or in the BRAF homolog RAF1. So even back then, and there was a whole series of papers at that time, we already started thinking about this potentially being a very interesting target for therapies. There were two things that we didn't know at that time, or three things. One of them was, what are the remaining 15%? Would they also have a hit in the same pathway? The second question was, might there be a collaborating pathway that is important that we have overlooked so far? And the third question was, would really a single oncogenic hit in one of these genes be sufficient to drive tumorigenesis in these tumors? What we also have appreciated before, and now when looking at a lot of unpublished or summarized data in a meta-analysis that David Jones, together with the CEO of low-grade glioma preclinical working group, has put together, that these different hits in the pathway are not equally distributed across the different anatomic locations of these tumors. So for instance, in the optic nerve, you have less than 50% presence of BRAF fusions, whereas in the cerebellum more than 3/4 are fusion positive. So, and another thing that came out from this meta-analysis, which at this stage can just be called interesting, but we don't know the relevance yet, is that even the different fusions in BRAF might be associated with different outcome. We know that they have some bias in terms of where they occur, in terms of anatomic compartment, but even independent of that, they seem to have slightly different biological function, and it will be very interesting to further study that. So then the next questions, as I said, is the single hit sufficient to cause tumorigenesis? What are the remaining 15%, or can they be explained with a MAP kinase hit as well? So the first question was kind of addressed by a study that was done by Jan Gornisch in Peter Lichter's group in Heidelberg, where he has introduced in the ARCAS system different variants of BRAF wild type, and including also the V600E mutation that we know. And it was really striking that with a very high penetrance and quite a short latency, which was surprising in the beginning to us, he could generate tumors in a mouse model, which also faithfully recapitulate pilocytic astrocytoma, not only because the histology looks similar, but also because of the low proliferation rate, which we know is happening there. And one of the interesting features in that model, and this seems to be something that is going in parallel with patients, is that these tumors in the beginning tend to grow very fast, which might be the tumors that we detect early on as pylomyxoid astrocytomas, and then they basically stop proliferating at some stage, and they basically stay at almost the same size for months and months. So the mice don't die from their tumors because at some stage these stop growing, which is basically something or phenomenon that we know as oncogene-induced senescence. The next question was, so what about the remaining 15%? And this was basically addressed by doing whole genome sequencing in different places in the world, and I'm just presenting your data because there are the pictures already made, but I could have equally presented the St. Jude's data here. Basically it's all about MAP kinase signaling. We identified more different mechanisms to cause BRAF fusions, two of these examples are shown here, even involving genomic translocations rather than focal aberrations on chromosome 7. We also identified mutations in other MAP kinase genes, such as PDPN11 and FGFR1. And one really interesting feature of all of that was that the ligand that is needed for FGFR1, which is one of the receptor tyrosine kinases at the cellular membrane to be activated, is FGF2, and this is something that is really unique to the expression profile of the pilocytic astrocytomas, in contrast to all higher grade astrocytomas, that FGF2 has by far the highest expression. So we think that probably for pilocytic astrocytoma, the receptor tyrosine kinase that is getting activated with this FGF2 FGFR1 pair. So in summarizing that, and again, this was two studies that were published back to back and independently came to that conclusion. In pilocytic astrocytoma, we find 100% hits in the MAP kinase pathway at different level, and this is just extending the slide that I showed before, and the same is true for the pilocytic astrocytomas in the St. Jude cohort. So here we have included pilocytic as well as higher grade, so this is kind of a mixture of cases where we have some other mutations over here, which what will I refer to later on. But the other thing that is not shown here is that none of the two groups was able to identify any other recurrently mutated gene or recurrently mutated pathway outside of MAP kinase signaling, really stressing the fact that this might indeed be a single pathway disease in its true sense. And I'm sure that Mark and Chuck will basically refer back to that, because this is the basis for really being an ideal candidate for targeted drugs. So now, when thinking about some of the puzzles that we still have, one of them being that obviously these anti-RK fusions that we also found as a hit in a proportion of pilocytic astrocytomas, which also feeds into the MAP kinase pathway, can also be identified, especially in infant glioblastomas. And it will be really interesting once we have larger cohorts to look after the commonalities and differences between an anti-RK mutated low-grade astrocytoma or polycytic and an anti-RK fused infant GBM. Whether these are the same, whether these are similar, we still think that they are really two different entities, but it's interesting that really grade 1 and grade 4 meets there through this mutation. We have similar examples such as the V600E mutation in BRAF, where we know that it can occur across all the different grades of glioma, so it's not completely surprising. So what about the other low-grade gliomas? And I think that one of the most important messages, and I will repeat that at the end again, that I just try to get across today, is that really low-grade glioma is something that we should probably get rid of at some stage, because we really don't want to talk about all of them in one basket anymore, but rather appreciate that we are talking about 10 or 15 different diseases. One of the kind of very nice and elegant additions to the pilocytic sequencing studies was several papers that indicated that both MYB and MYB-L rearrangements in lower grade astrocytomas, and that was shown already about 4 years ago, and then again in 2013, and also turned up again in the sequencing paper from Senchu that I just presented, that really these MYB and MYB-L rearrangements seem to be quite specific for grade 2 astrocytomas rather than polycytic astrocytomas. We also think that the FGFR fusion as well as mutation has some overlap between grade 1 and grade 2 tumors. So again, it will be interesting to collect more of these, since this is also a potential drug target, whether they are really different or whether this is just one class of either grade 1 or grade 2 that we should probably call according to the presence of a druggable target rather than the histological grade. What about all the other much smaller entities defined by histopathology? So one of the attempts that we have chosen for low-grade gliomas as well as other brain tumor entities was DNA methylation fingerprinting. The reason for that was already discussed this morning, is that really this promoter methylation that we get when using promoter methylation arrays is rather reminiscent of a fingerprint of the cell of origin, that it being reflecting somatically acquired epigenetic changes. So this is something that remains very stable across the disease from the cell of origin until a relapse of a tumor. And we have collected a large number of low-grade glioma samples. Overall, it's about more than 400 tumor samples, and also 80 normal samples from different locations in the brain. It contained sufficient numbers of all the entities that are mentioned here, which are pathologically defined. And as I said, it includes 80 samples from different normal brain regions. And we also added some glioblastomas and the different groups of glioblastomas, because we more and more see these problems sometimes in the diagnostic setup between lower grade and higher grade tumors, where one pathologist calls it a lower grade and the other one calls it a GBM. So for us in the clinic, and we will all have experienced that, it's very difficult to make a call here. Do we take the low grade and tell the patient, oh, you're going to be fine no matter what we are doing, or do we take the high grade and say, oh, you're going to die within the next 12 months? So this is how this large clustering looks like now, taking all these different glial tumors plus the normals into account. And I will just not go through all of the different groups with you, but one thing, one kind of major split that you can see is the one between cerebellar and midline tumors and structures, and the hemispheric on the other side. So this makes the biggest split in the methylation fingerprinting. But you can also appreciate that, for instance, if you take different entities that were defined by pathologists, such as ganglioma or pilocytic in the cortical region or DNET, they can be appreciated and acknowledged here as subtly different entities. So in trying to interpret and summarize that for you, we see this major split with the localization, but within the large kind of anatomic compartments, there seems to be a subgrouping that justifies also from a biological perspective the distinction of several entities. And if you go through the different compartments now, to start with the hemispheric tumors, the angiocentric gliomas also, this is a very rare disease, seems to be a very clearly distinct biological group. The DNETs, gangliogliomas, and cortical PAs, also they are very similar to each other. They can be distinguished by the methylated fingerprinting. We think that the pleomorphic centroastrocytomas are basically the main problem distinguishing low grades from high grades, because some of them end up as low grades, some of them end up as high grades, and sometimes it's just to the discretion of the pathologist saying, oh, this looks a little bit more anaplastic, so let's put it in the high grade group, or this doesn't have too much anaplasia, so let's put it in the low grade group. So really, here we have evidence that all of them are the same, but they certainly have a worse prognosis than a pilocytic astrocytoma, but they also have a better prognosis than GBM. And the third feature about them is that most of them have BRAF V600E mutations, so we think that it really justifies to take them as a group and treat them separately. In the posterior fossa, we clearly have this low-grade astrocytoma group, which in the posterior fossa is clearly dominated by pilocytic astrocytomas. We also have first evidence, and we need more samples to really confirm that, for biologically different entities in terms of gangliocytoma and rosette-forming neural tumors of the fourth ventricle, but here we need more cases. And then when talking about the midline tumors, again, these low-grade astrocytomas are mostly PAs. We clearly don't see a biologically justifiable subgroup of pilomyxoid astrocytoma. We think that they only have a slightly worse prognosis because they tend to occur in the midline, they tend to be not accessible to surgery, and this is why what explains the difference, but clearly biologically they are basically the same. And again, some of these smaller entities, such as the diffuse infantile gangliogliomas, the choroidogliomas of the third ventricle, or clearly also the CGAS, they seem to be clearly different as an entity. So, as you can tell from that, we really have to appreciate that low-grade glioma is not all the same. And we now, once we start understanding the molecular biology, and especially when it comes to targeted therapies, we really have to take that into account. So, in conclusion, I think that I hope that we can convince you that really pilocytic astrocytoma is a disease of the MAP kinase pathway. We find 100% usually mutually exclusive mutations in that pathway in this disease. Yet, and I'm sure that Mark and Chuck will talk about that later, we don't have the magic treatment for them in terms of targeted drugs, but I think that we are in a really good way. For the Astros grade 2, they really have to seem to have kind of hallmark genetic aberrations that are specific to these, and it will be interesting in the clinical trial cohorts to check whether this is also associated with groups that have inferior prognosis independent of surgical accessibility, and certainly the FGFR mutants are also an attractive target for therapies. IDH mutations are extremely rare in pediatric glial tumors, but of course they exist sometimes, especially in the adolescents. And we are pretty confident, I think that this is true for adult as well as pediatrics, that there's no such thing as an oligoastrocytoma. So either it's an oligo or it's an astro, and it's just sometimes hard to tell from the morphology. but if you add the genetics and the biology, then for every one of them it's pretty clear what they are. And in the group of the others, we think that some of the histological entities that have been defined in the WHO classification, these seem to appear dispensable. So for instance, the palomyxoid or some other variants of grade 2 astrocytomas. Some of them clearly seem to have a distinct biological profile which justifies to really understand them. Maybe they have completely different driving mechanisms. And this group of pleomorphic xanthoastrocytomas, really in terms of its methylation fingerprinting, and usually they also have homozygous deletion of 9p21, which is the locus of p15 and p16. They seem to be more closely related with GBMs or higher grade gliomas, but in any case should be treated as a separate entity. And just to finish off with two of the take-home messages for today. One of them is that I think that we shouldn't think about running low-grade glioma trials without thinking about biology and subgroups anymore. So this is clear from many studies that were done now, that they seem to have different cells of origin or different times of malignant transformation. And of course, and this is probably the most important thing, is that molecular characterization should be part of routine diagnostics. It's no longer acceptable to call something LGG NOS and you don't know about BRAF status, you don't know about anything else. It's equally important to get this information in a routine practice than we do getting the pathological information. And I think one of the most important arguments is that we are already starting to move forward with these targeted drugs. And we cannot all of a sudden, when Mark tells us, so now the MEK inhibitor XYZ is ready for prime time to apply it worldwide, we cannot all of a sudden then think of establishing the infrastructure that is needed to do the molecular testing. We have to introduce it now to be ready for prime time. With this, I want to appreciate the CO low-grade glioma preclinical working group, which I'm very pleased to organize together with Olaf Witt. Of course, my group at the German Cancer Research Center, many collaborators throughout the world, and of course our funding agencies, without whom this work would not be possible. Thanks very much.